Hello students, welcome to today's program. The objectives of the topic will be to discuss the process of mitosis and to teach the process of meiosis. Now let us come first to mitosis and meiosis. In both unicellular and multicellular eukaryotes, cellular reproduction is a cyclical process of growth, mitosis, that is nuclear division, or what we call as karyokinesis, and usually but not always cell division, that is cytokinesis. The cycle of growth, mitosis and cell division is called the cell cycle. In proliferating somatic cells, the cell cycle consists of two phases, the mitotic or division or M phase and an interphase between divisions. Interphase consists of three stages, that is gap 1, S and gap 2 phases. During the pre-synthesis stage, that is the gap first, the cell prepares for DNA and chromosome replication, which take place in the S stage. In the post-synthesis stage, that is gap 2, the cell prepares for cell division or the M stage. In other words, chromosome replication takes place in interphase and then mitosis occurs, resulting in the distribution of a complete chromosome set to each of the two progeny nuclei. The orderly distribution of duplicated chromosomes in a mother cell to its daughter cell is the essence of mitosis. Each chromosome in a mother cell is duplicated prior to the onset of mitosis, specifically during the S phase. At this time, individual chromosomes cannot be identified because they are too extended and too thin. The network of thin strands formed by all the chromosomes within the nucleus is referred to as chromatin. During mitosis, the chromosomes shorten and thicken. That is, they condense out of the chromatin network and individual chromosomes become recognizable. After mitosis, the chromosomes decondense and the chromatin network is reformed. Mitosis occurs in both haploid and diploid cells. It is a continuous process, but for purpose of discussion, it is commonly divided into four stages, that is prophase, metaphase, anaphase and telophase. Let us discuss these phases. First, let us come to prophase. In prophase, the chromatids condense, so they gradually appear shorter and fatter under the microscope. By late prophase, each chromosome which was duplicated during the preceding S phase of the interphase can be seen to consist of two sister chromatids. While condensation is occurring, the nucleolus shrinks and eventually disappears in most species. Now metaphase. The nuclear envelope breaks down at the end of the prophase, denoting the beginning of prometaphase. The developing spindle now enters the former nuclear area. A specialized multiprotein complex called a kinetochore binds to each centromere. The kinetochores are the sites for the attachment of the chromosome to spindle microtubules known as kinetochore microtubules. During metaphase, the kinetochore microtubules orient the chromosomes so that their centromeres become aligned at the metaphase plate, a plane halfway between the two spindle poles, with the long axis of the chromosome oriented at 90 degrees to the spindle axis. Coming to the anaphase. During anaphase, the joined centromeres of sister chromatids separate, giving rise to two daughter chromosomes. Once the paired kinetochores on each chromosome separate, the sister chromatid pairs undergo disjunction, that is separation, and the daughter chromosomes move towards the opposite poles. In anaphase, the daughter chromosomes are pulled towards the opposite poles of the cell by shortening microtubules attached to the kinetochores. As they are pulled, the chromosome assume characteristic shapes related to the location of the centromere along the chromosome's length. The movement continues until the separated daughter chromosomes have reached the two poles, at which point chromosome segregation has been completed. Cytokinesis usually begins in the later stages of anaphase. Now lastly, the telophase. At the start of telophase, the two sets of daughter chromosomes are assembled into two groups at opposite ends of the cell. The chromosomes begin to uncoil and assume the elongated state characteristic of interphase. A nuclear envelope forms around each group of chromosomes. The spindle microtubules disappear and the nucleolus form. At this point, nuclear division is complete and the cells now have two nuclei. Now coming to another topic, that is meiosis. Meiosis is the two successive divisions of a diploid nucleus after only one DNA replication, that is chromosome duplication cycle. The original diploid nucleus contains one haploid set of chromosomes from the mother and one set from the father. With the exception of self-fertilizing organisms, for example, many plants in which both sets of chromosomes come from the same parent. Meiosis occurs only at special point in an organism's life cycle. In animals, it results in the formation of haploid gametes, that is, eggs and sperm by gametogenesis. In plants, it results in formation of haploid meiospores by sporogenesis. A meiospore undergoes mitosis to produce a gamete-bearing multicellular stage called the gametophyte. Before meiosis, the DNA that makes up homologous chromosomes replicates, and during meiosis, these chromosomes pair and then undergo two divisions, meiosis first and meiosis second, each consisting of a series of stages. Meiosis first results in reduction in the number of chromosomes in each cell from diploid to haploid, that is reductional division. Each resulting pair of attached sister chromatids count as single chromosome. And meiosis second results in the separation of the sister chromatids. As a consequence, each of the four nuclei that come about from the two meiotic divisions receives one chromosome of each chromosome set, that is, one complete haploid genome. In most cases, the division are accompanied by cytokinesis, so the meiosis of a single diploid cell produces four haploid cells. Meiosis first, in which the chromosome number is reduced from diploid to haploid, consists of 5 stages: prophase first, prometaphase first, metaphase first, anaphase first, and telophase first. Now starting with prophase first. When prophase first begins, the chromosomes have already duplicated, with each consisting of two sister chromatids attached at a centromere. Prophase first is divided into a number of the substages. Prophase first of meiosis is similar to prophase of mitosis. The important difference between prophase first of meiosis and prophase of mitosis is that homologous chromosomes pair with each other in meiosis and crossing over occurs only in meiosis. In leptonema, that is early prophase, the leptotin stage, the extended chromosomes begin to condense and become visible as long, thin threads. Once a cell enters leptonema, it is committed to the meiotic process. In zygonema, early to middle prophase first, that is the zygotine stage, the chromosomes continue to condense. The homologous pairs of chromosomes actively find each other and align roughly along their lengths. Each pair of homologous then undergo synapses, that is the formation along the length of the chromatids of a zipper-like structure called the synaptonemal complex. The synaptonemal complex aligns the two homologous precisely, base pair for base pair. The telomeres of the chromosomes play a key role in the initiation of synapses. That is, during meiosis first, telomeres are clustered on the nuclear envelope to produce an arrangement called a bucute. Because of its resemblance to the stems from a bouquet of cut flowers. In some way, the telomeres move the chromosomes around so that homologous chromosomes align and undergo synapses. Now the pachynyma, that is the middle prophase first, or what we call as the Pachytine stage, starts when a synapsis is completed. Because of the replication that occurred earlier, each synapsized set of homologous chromosomes consists of 4 chromatids and is called a bivalent or a tetrad. During pachynyma, a most significant event for genetics occurs, that is the crossing over. The reciprocal physical exchange of chromosome segments at corresponding positions along pairs of homologous chromosomes. The positions at which crossing over occurs along the chromosomes are largely random and vary from one meiosis to another. The physical exchange that occurs in crossing over is facilitated by the alignment of the homologous chromosomes brought about by the synaptonemal complex. If there are genetic differences between the homologous crossing over can produce new gene combinations in a chromatids. There is usually no loss or addition of the genetic material to either chromosome, since crossing over involves reciprocal exchanges. A chromosome that emerges from meiosis with a combination of alleles that differs from the combination with which it started is called a recombinant chromosome. Therefore, crossing over is a mechanism that can give rise to genetic recombinations. At the end of the pachynyma, the synaptonemal complex is disassembled and chromosomes have started to elongate. In diplotynyma, that is the middle to late prophase first, or what we call as the diplotyne stage, the synaptonemal complex disassembles and the homologous chromosome begin to move apart. The result of crossing over becomes visible during diplonyma as a cross-shaped structure called chiasma, plural chiasmata. At each chiasma, the homologous chromosomes are very tightly associated. Because all four chromatids may be involved in crossing over events along the length of homologous, the chiasma pattern at this stage may be quite complex. In diakinesis, the late prophase first, the chromosome condenses even more, making it now possible to see the four members of the tetrads. The chiasmata are clearly visible at this stage. Now coming to metaphase first. In prometaphase first, that is early metaphase, the nucleoli disappear. The nuclear envelope breaks down and the meiotic spindle that has been forming between the separating centriole pairs enter the former nuclear area. As in mitosis, kinetochore microtubules attach to the chromosomes. That is, kinetochore microtubule from one pole attached to both sister kinetochore of one duplicated chromosome, and kinetochore microtubules from the other pole attached to both sister kinetochore of the other duplicated chromosome in a tetrad. Non-kinetochore microtubules from each pole overlap in the center of the cell. Metaphase first. In metaphase first, the kinetochore microtubules align the tetrad on the metaphase palate. Importantly, the pair of homologous, that is the tetrads, are found at the anaphase palate. In contrast, in mitosis, replicated homologous chromosomes, that is sister chromatid pairs, align independently of one another at the metaphase palate. Now the anaphase first. In anaphase first, the chromosomes in each tetrad separate, so the chromosomes of each homologous pairs disjoin and migrate towards opposite poles, the area in which new nuclei will form. At this stage, each of the separated chromosome is called a dyad. This migration assumes that maternally derived and paternally derived chromosomes segregate randomly at each pole. Of course, spots of chromosomes may have exchanged during the crossing over process, and that at each pole there is a haploid complement of replicated chromosomes with associated sister chromatids. At this time, homologous chromosomes have segregated from each other, but sister chromatids remain attached at their respective centromere. In other words, a key difference between meiosis first and mitosis is that sister chromatids remain joined after metaphase in meiosis first, whereas they separate in mitosis. Now, the telophase first. In telophase first, the dyads complete their migration to opposite poles of the cell and the spindle disassembles. In some species, but not all, new nuclear envelopes form around each haploid grouping. In most species, cytokinesis follows telophase first, producing two haploid cells. Thus, meiosis first, which begins with a diploid cell that contains one maternally derived and one paternally derived set of chromosomes, ends with two nuclei, each of which is haploid and contains one mixed paternal set of diodes. After cytokinesis, each of the two progeny cells have a nucleus with a haploid set of diodes. Now coming to meiosis second. No DNA replications occur between meiosis first and meiosis second. Meiosis second is similar to mitotic division. In prophase second, the chromosomes condense and a spindle forms. In prometaphase second, the nuclear envelope, if formed in telomere first, breaks down and the spindle organizes across the cell. Kinetochore microtubules from the opposite poles attached to the kinetochores of each chromosome. In metaphase second, the movement of the kinetochore microtubules aligns the chromosome on the metaphase plate. During anaphase second, the centromeres separate and now daughter chromosomes are pulled to the opposite poles of the spindle. One sister chromatid of each pair goes to one pole and the other goes to the opposite pole. The separated chromatids are now considered chromosomes in their own right. In the last stage, that is the telophase second, the chromosomes begin decondensing. A nuclear set of chromosomes and cytokinesis takes place. After telophase second, the chromosomes continue condensing, eventually becomes invisible under the light microscope. The end products of two meiotic division are four haploid cells, what we call as gametes in animals, from one original diploid cell. Each of the four progeny cells has one chromosome from each homologous pair of chromosomes. Because of crossing over, these chromosomes are not exact copies of the original chromosomes. There is an interrelation between the function of genetics and the structure of the cell. Ultimately, it is the genetic material of an organism which determines how cell works. Genetic material or DNA is the blueprint from which the information regarding the cell structure and function is taken or transcribed in the form of mRNAs, and then these RNA molecules are translated on ribosomes, the protein factories. Finally, the proteins determine the structure of the cell by acting as enzymes, building blocks, or what we call as hormones. Now lastly, meiosis and Mandela's laws. When traits are passed from one generation to another, they follow principles of genetic inheritance that were first defined by George Mendel. Mendel's studies yielded three laws of inheritance: the law of dominance, the law of segregation, and the law of independent assortment. Each of these can be understood through examining the process of meiosis. Meiosis: when a cell duplicates its DNA and divides twice to produce four gametes or reproductive cells, the process is called meiosis. Most cells in the body are diploid, meaning they have two copies of each chromosome. But because gametes have gone through meiosis, they have one copy of each chromosome and are called haploid. During sexual reproduction, two gametes called the egg and sperm join together and form a diploid cell that will eventually become an individual organism. This diploid cell, called a zygote, received one copy of each chromosome from each parent. The appearance or phenotype of the new individual will depend on whether it inherited recessive or dominant copies of various alleles from its parents. Variant copies of the genes are called alleles, and since plants and animals are diploid, they have two alleles for each gene. The law of dominance. A dominant trait is a trait whose appearance will always be seen in offspring. In other words, dominance describes the relationship between two alleles. If an individual inherits two different alleles from each of its two parents and the phenotype of only one allele is visible in offspring, then that allele is said to be dominant. Mendel's law of dominance states that if one parent has two copies of allele capital A, the dominant allele and the second parent has two copies of allele small a, the recessive allele, then the offspring will inherit a capital A small a genotype and displaying the dominant phenotype. Now the law of segregation. A parent may have two distinct alleles for a certain gene, each on one copy of a given chromosome. Mandela's second law, the law of segregation, states that these two alleles will be separated from each other during meiosis. Specifically, in the second of the two cell divisions of meiosis, the two copies of the chromosome will be separated from each other, causing the two distinct alleles located on those chromosomes to segregate from one another. Now the law of independent assortment. Mandela's third law, the law of independent assortment, states that the way an allele pair gets aggregated into two daughter cells during the second division of meiosis has no effect on how any other allele pair gets segregated. In other words, the traits inherited through one gene will be inherited independently of the traits inherited through another gene, because the genes reside on different chromosomes that are independently assorted into daughter cells during meiosis. Thank you. This was all from today's lecture. Hope you have understood this lecture very well. Thank you.